I know many of you are observing Ramadan, and late morning to early afternoon is a hard time physiologically, regardless in our bodies. A lot of things are cycling downward, so I understand if you have to put your head down, or let me know if there's any way I can assist as you move through this month of reflection, but also I know that it's a little bit harder physiologically too, so I am here to support you all. All right, so we left off talking about exergonic and endergonic reactions, reactions where you need an input of energy or you release energy. And what happens with these kinds of reactions is that they are coupled together within our cells. One of the just so many amazing things about the way our cells run. This is one of them, that you have one reaction that happens, and it is an exergonic reaction where it gives off energy, and then you have an endergonic reaction over here that takes in that energy, and then it fuels that reaction to run. And these reactions are constantly fueling each other. So the need for, is one of the characteristics of life, the need for organization of living things. It is just amazing how these reactions will fuel each other, and not just one fuels the other, but then that one will fuel the next one, the next one, and the next one, and the next one, and the next one. All right, so energy, remember, chemical bonds, when you have a bond, that means that you have stored or passive energy. When you break a bond, it releases energy. We are going to see certain compounds that are broken down that will, when they are broken down and the bonds are broken, they release energy to fuel something else, or there might be two compounds that are waiting and they're going to make a bond and store energy from a reaction, from an exergonic reaction. So what we call these compounds are energy carrying molecules, because they are going to take energy in and they are going to form a bond. We have some that we will see in many different places as we look more closely at photosynthesis and cellular respiration. So let's talk about ATP first. ADP, so we have ADP, which is adenosine diphosphate. And we have ADP, which is adenosine diphosphate, plus a phosphate, make ATP, adenosine triphosphate. So I like to write it out like this so that you can see the di and the tri. Di means two. That when we're looking at adenosine, it has two phosphates on it. That's what the di stands for. Adenosine with two phosphates. We're going to add another phosphate. the other things that are implied in this equation are that we're also going to have to add in 2 electrons. And those 2 electrons, when they come together, it's the sharing of a pair of electrons, which makes a covalent bond. The other thing that we need is we need an input of energy. And that input of energy is going to be from an exergonic reaction that is coupled to this reaction. We get an input of energy from an exergonic reaction coupled to this energonic reaction. So when we're talking about these electrons, this covalent bond that is formed here is made up of 2 electrons, plus there is energy held in this newly formed covalent bond from the exergonic reaction that's coupled to this. This reaction of ADP plus P to make ATP also serves as an exergonic reaction often. So when it goes in this direction, and you take your adenosine, and let me point out, adenosine tri, meaning 3, you have 3 phosphates on your adenosine. So adenosine triphosphating, 1, 2, 3 phosphates. And a lot of times once this is made up and it's built up, you can break this bond and send this in this direction. So it becomes an exergonic reaction that lets go of 2 electrons, releases energy, becomes ADP plus P. So that when it goes that way backward, it's an exergonic reaction, but when it goes this way forward, like we've been mapping out, it's an energonic reaction. So when it goes in that direction, and we break off that surge phosphate, and we break the bond, so when we do that, when we break this bond to take off that third phosphate, then we have this whole thing that happens where you get your ADP here, you get your P, we get from this two electrons released from the breaking of that covalent bond. So just again, when we break this bond here, you get your ADP. You get a phosphate alone. When we break this covalent bond, which is made up of the sharing of a pair of electrons, we release two electrons from breaking that covalent bond. And in this covalent bond also, you had some energy that was stored in that bond. As we go through cellular respiration and photosynthesis, we will see that sometimes we use this exergonic reaction, and sometimes we make ATP as an endergonic reaction. So ATP just basically, a lot of times in our cells, it gets broken down and it releases energy, and then the same parts get used and built back up. So it is very often going from that to that, that to that. And we're using the same parts over and over again, but we need new inputs of energy every time. Because if you remember back from the laws of thermodynamics, that energy goes down and down and down as it gets used. So you need more inputs of energy. But the same parts, the ATP and the ADP plus P, they just get used over and over again. So this is, today I want to introduce one other energy carrying molecule, which is NADPH. So let me, all right, so we have an NADPH. So we have that happens also. We have an NAD plus an H, and they make NADP, and we add on that H. here, we're using 2 electrons to add on that hydrogen to the NADP. This is an endergonic reaction, which needs an input of energy from an exergonic reaction. So input of energy gets stored in that bond. So same principles apply, just a different energy carrying molecule. We'll introduce one more as we get into cellular respiration also. So we're going to walk through ATP again because it's a very important energy carrying molecule. So subunits of ATP, adenosine triphosphate. Remember the T, the tri, is because we had 3 phosphates added on to our adenosine, and they make ADP plus P. We just said that ATP, that when the last phosphate breaks off, it can release energy. So it can function as an exergonic reaction, or it can take in energy and function as an endergonic reaction. This really, really, really important energy carrying molecule, ATP, will fuel so many things in our body. All right, so what is this? The breakdown, when we break ATP down, which of these reactions is it? So the one that's breaking down. Okay, it's an exergonic reaction. It breaks and releases energy. Energy is released or exits from that bond that is broken. So here's a little nicer version of what I wrote on the board. You've got your adenosine and your 1, 2, 3 phosphates. So this is ATP, adenosine, triphosphate. When we break this bond apart, we release energy. I always like to put the electrons in. You also get 2 electrons released. So release of energy, 2 electrons released. This bond was broken, so we have ADP, 2 phosphates, and that other phosphate by itself. Now, in the bigger picture of all of this is really what's happening in our cells, is that within our cells, you have, when we break down glucose, here's the thing about energy in our cells, is that glucose, or you really can think about that as any kind of food. So any of our food, it's valuable, right? We spend a lot of our time working to buy food. when we didn't have jobs, when we were, like we go back 10,000 years, our ancestors spent a lot of time hunting for food because we need energy, food sources for survival. So our food sources, which are really, really valuable, now let's go all the way down to the cellular level. If you have like a nice sandwich, is that significant to your cells in terms of, can your cells eat that sandwich? No, it's like too big, right? And so we have this whole digestive system that you chew it apart, and then the food goes into your stomach, and it goes into your intestines, and then the intestines bring it into the broken down materials into your blood, brings it out to the cells, and the cells do what's called cellular respiration to break down our food into smaller bits of energy that our cells can use. So the way our body works, and in terms of the cellular level, is that while food is really, really important to us, it does have to go through many, many, many steps to get to something that's more valuable in our body. So when we break down glucose, and you have this whole process of glucose plus oxygen, when oxygen is present while we are breaking down our food, we can get a lot of the energy given off to make a lot of ATP. That's what we call aerobic cellular respiration. Aerobic means with oxygen. So that when we use oxygen to break down our food, we can transfer that energy into a lot of ATP. And ATP is what our cells can use to fuel all the things that they have going on. They can't use that sandwich, but they can use the ATP that eventually is the energy that came from that original sandwich. So glucose getting broken down by cellular respiration, we give off carbon dioxide, that's the respiration part, use oxygen, give off carbon dioxide, just like how we breathe in oxygen, breathe out carbon dioxide. We have a cellular respiration that happens when we break down our food with oxygen. We give off carbon dioxide, water, and heat energy, but that is coupled to an ADP plus a P, and they're waiting for the energy given off from the taking in of our larger food to create ATP, which is better used by our cells. So there's where kind of the grand scheme coupling happens. And then ATP can be used in our cells, that when we have, if you remember monomers and polymers from some of our biomolecule groups, that one biomolecule group was the proteins, and the proteins do a lot of really important things in our body, so that when you have something like the monomers, the amino acids have to form a bond to make up important things in our body, that when you take that ATP that captured the energy from the breakdown of our food, you can break that bond and release that energy into making important things in our cells, like proteins. But this could be anything. This could be the monomers of fats. This could be the monomers of carbohydrates. So we can just be, anything of building up comes from the energy released in the breaking down of our food, captured by ATP. When we break down ATP, that energy fuels something else. So pretty well organized and orchestrated within our bodies. So let's think about this. When a high energy bond of ATP is broken down, what primarily happens to the released energy? Is it lost as light energy? Can we lose light energy? It functions as a second messenger. That sounds weird. It polarizes the cell. It converts inorganic phosphates into energy carrying molecules. you might be like, maybe that sounds like I heard something about phosphates and energy, right, and carrying molecules. Okay, that sounds complicated. Let's read this one. It drives endergonic reactions in the cell. Yeah, okay, good. So while that fourth one was a little like, oh, this is where the amount of studying you do, it'll get you to these two, right? Because you'll go, no, these are ridiculous, right? So just by sitting in class, you can go, no. And then you get to these two. And you're like, oh, did she say stuff? She said words like this. And then you might be like, but this sounds more familiar. And then what we do when we haven't prepared enough on exams is we go, I think it's fine, but no, I'm gonna go with this one. And then you second guess yourself, right? Okay, so grander scheme, like if we go bigger than just ourselves, the process of photosynthesis takes energy from the sun. Here's an input of energy. So in the big scheme of energy, we have an endergonic reaction. You have sunlight energy enters into the process of photosynthesis. The process of photosynthesis gives off oxygen and also makes sugar, but really it makes food, right? When we are talking about plants, they can make oils and fats and beans and vegetables and fruits and nuts and seeds and grains. It can make all kinds of stuff. And these, what I like to just replace this with is the idea of food that is made by plants. When animals like us eat the food and breathe in oxygen, we give off energy, we make ATP, we give off carbon dioxide. Carbon dioxide goes back to the plant. They take that carbon dioxide with the sunlight energy and water, and then they go through it all again. And so here we have an endergonic reaction fueling an exergonic reaction, fueling an endergonic reaction, fueling an exergonic reaction, so that these processes of cellular respiration, us breaking down our food, comes from photosynthesis, and photosynthesis fuels us to break down our food, which then we give back to photosynthesis our carbon dioxide, and they need a new input of energy, and then we keep getting more food by some of these things like the molecules of the oxygen, the parts of maybe like carbons and things like that in our food. Those get all recycled and stay in, but we have to keep getting an input of energy so that the laws of thermodynamics are saying that while photosynthesis can fuel cell respiration and the parts of cell respiration can fuel photosynthesis, you still, with every time around, you need a new input of energy. That we need that renewable source of energy every single time. So, as I said, there's a lot of orderliness to all of this. That's one of the characteristics of everything being living, is that we are super duper organized. And what happens is not just one exergonic fuels an endergonic, but you have coupled reactions that happen over and over and over and over and over and over and over. So we have what are called metabolic pathways. And we're going to see there are many steps that you're going to build this up. And then you're going to break it down. And energy is going to be released. And then that's going to go in some other metabolic pathway. And we're going to continue on this metabolic pathway. But we always have these other metabolic pathway offshoots as well. So there's a lot of steps going on. There's where that organization needs to take place. Each step gets what we call catalyzed by an enzyme. Catalyzed. It's facilitated, helped. That enzymes are going to come in here, and while these reactions take place, the enzyme, what they're going to do is they're going to help to make it better. So let's say you're having a party, and that maybe you don't have a lot of money, and you have just enough money to maybe buy food and beverages and you can have friends and family over, but you have to do all the cooking and all the organizing. You have to go out and do all the shopping, right? It's going to take a lot of energy. You'll get it done though. But what if you could hire a party planner? Wouldn't that be nice? You're gonna have a party and you just pay someone and they organize everything. You're going to use a lot less energy with the party planner as opposed to having to do it all yourself. Even though it's cheaper, you're gonna use a lot of energy. So we can kind of do one of those two things in our cells, is that our cells can just kind of try and wait for different compounds to bump into each other and react, and that will take a long time and a lot of energy for the molecules they're bouncing around, and eventually they run into each other, and it's going to take a lot more time and energy. Or we can hire an enzyme for each reaction. And what the enzyme is going to do is the enzyme you put an enzyme into this reaction, and the enzyme is going to say, hey, NADH, hey, hydrogen, come here. 2 electrons, come here. They're going to, the enzyme is going to get some energy, and it's going to be like, all of you 4, come here, and put them together to make NADPH quickly. And when that's made, this enzyme can be like, okay, I got that one through, next. NADP, H, 2 electrons, energy. Okay, got another one. Let me do another one. Let me get another group. And they can facilitate getting all of these things together quickly in one place to make the compound that is needed elsewhere in the cell. But without having the enzyme, what the cell would have to rely on are these 2 within the cell just bumping into each other, while also 2 electrons bumped into them and an input of energy bumped into them. And the likelihood of this happening quickly is not very good. So the enzymes, when you stick an enzyme into a reaction and they catalyze, they can bring everybody together quickly and make that product quickly. So that's what we mean by the enzymes speed up the reaction, because they can quickly bring everyone together. Otherwise, it just takes a really long time for everything to just organically, accidentally bump into each other within the cell. We're going to look closely at 2 really, really important metabolic pathways: cellular respiration, the way that we break down our food, and photosynthesis, the way that our food is made. Why is photosynthesis considered an endergonic reaction? Is it because activation energy is not required? Did we talk about activation energy yet? No, right? Photosynthesis does not comply with the physical laws of the universe. Sugar has less energy than the sun. Protein catalysts are not needed. Low energy reactants are converted into high energy products. That sounds the best. That we have a bunch of carbons from carbon dioxide, and they go through a series of reactions where those low energy carbon dioxides become our food. So we have low energy reactants are converted into that energy from the sun, takes 6 carbons and carbon dioxide and it puts them together to make glucose. And then glucose can go into other metabolic pathways to become other kinds of carbohydrates, other kinds of proteins, other kinds of fats, or foods that have a combination of proteins, carbs, and fats. All right, so substrate. Now, when we use an enzyme, remember that just like basic chemical reactions, we said this and this are called the reactants, and the thing at the end is called the product. But when an enzyme catalyzes a reaction, we're going to change the name of these, and we're going to call the reactants substrates. So a substrate is a reactant when we're using an enzyme to facilitate or catalyze the chemical reaction. it gets a fancy name because the enzyme is going to bind to the substrates. So the idea is that the enzyme provides a meeting place that is literally going to grab an NADP, grab an H, so that these two can fit together on the enzyme. So they're going to fit together, and they're going to fit onto the enzyme, so that the enzyme's like, come here, come here, come here. And the enzyme holds onto them and pushes them together with the electrons and the energy, and then it pushes them out. And so this is a meeting place that is going to grab these and push them through. We're going to grab those, and we're going to grab the electrons and the energy. They're going to make all four of these come together and push them out. So they are called then a substrate. We saw the grabbing on of substrates back in the last chapter when we took a look at carrier proteins, receptor proteins, and active transport. Remember, it grabbed it, pushed it through. Grabbed it, pushed it through. That we had that grabbing on, and what we're talking about is that when we're looking at these kinds of proteins, that they are often the enzymes to meet with the substrates and get the electrons and the energy and make that reaction happen. So like this, we saw that when we had this carrier, you had the binding of this substrate to the, in this case we'll call it an enzyme, and then made that reaction go through. And same thing here. Again, you've got the binding. And even when we're talking about active transport, when you had two binding sites, one for the molecule to be transported and for, remember? Now we put in ATP here. And we actually used the breakdown of ATP to fuel this going through. Okay, so there is always a little bit of activation energy. For any reaction to happen, you need to put in a little bit of energy. This energy is not, it's like aside from the energy that is captured by the bonds. So there's always just like a little bit extra to make reactions happen. And that's like kind of a general chemistry thing. So just to let you know that there is a little bit of energy that needs for the reaction to happen, but this is in addition to the energy that's captured in the bonds. All right, so let's go back to our enzyme substrate complexes, like basically what we have here is we put them all together in one place. And we form enzymes with our substrates, with our electrons, with our energies. This is what we would call an enzyme substrate complex. The place where the substrates go onto the enzyme is called an active site. It's where the enzyme actively grabs on. And so like behind this area, in front of the NAD, P and H, behind that would be a place where the enzyme actually grabs onto those substrates. The active site just is the place where they all get brought together close. And so one of the things that's so cool about the organization of our cells and the metabolic pathways and the reactions that happen is that everything is like pieces of a puzzle. That when you have your substrates come together and fit together like two pieces of the puzzle, just like what I indicated here. But also, our substrates are going to bind into our enzyme. And the reason why this is really important is that we have so much going on in our cells. We have millions of reactions that are happening within our cells simultaneously. So for this particular reaction, these two substrates fit perfectly in here, but you've got substrates for other reactions all over the place within the cell. So you can have some that are like that, and some that are like that. And what is happening is they're all like, can I get in there? Nope, nope, nope. But these are the only ones that can. And so this is a part of the organization, is the shape of compounds and molecules within our cells for binding sites or active sites allows the right substrates to pair with the right enzyme to make the right reaction happen. So just like pieces of the puzzle. So you will have in these metabolic pathways, we might have this reaction happens, and then a next reaction, and a next one, and a next one. And the shapes of these are different than the shapes of these, than the shapes of these, than the shapes of these. But every time you take two things and you put them together, substrate or enzyme, the shapes are specific to each kind of reaction. So, induced fit model. What that means is that when a substrate binds to its proper and correct enzyme, that it causes the enzyme to change its shape and push the reaction through. So we've seen this happen where our substrate binds into the binding site of the protein or the enzyme, and you can see it goes from being closed, it opens up, and then it goes back to its original shape. And this is what we mean by the induced fit model, that the enzymes will push things through and go back. And the only thing that will be pushed through is the thing that fits with the active site. So like this, for example, when these substrates, again, that we saw earlier, fit perfectly into the enzyme's active site, it makes these become something new, right? These two things became something new. Because the enzyme brought them together, made them react, added some electrons, added some energy, and took them from being that form into something at the end of the reaction. That our substrates became a product of the reaction. The enzyme, when it's grabbing onto them, it changes its shape, right? It goes like this, it's closed, grabs onto it, pushes them through, then it goes back. So that what happens is that it's temporarily going to facilitate or catalyze this reaction, and then it's going to go back to its original shape and be ready for new substrates that fit perfectly so that it can bring them together, make them react to become a new product, and it can go back and be used again. And the great thing that we save a ton of energy is that we don't, with every time you have substrates to make a new product, you do need new substrates, and you do need, and then you produce new product, and if you want to go again, you need new substrates, and you need new substrates. So you keep inputting that substrate, but you don't have to keep remaking the enzyme. Because the enzyme is just going to, in this case, it's going to sit there and it's going to grab these and get everybody to come together and then push them out. And then they're like, okay, I'm ready for more. Okay, 4 more, come. 4 more of you, come over. And the enzyme is just like a little machine that is being used over and over and over. So not only does the enzyme add to the organization that it can ensure that it has the right parts for this particular reaction, but it also, the enzyme saves us a lot of energy because it's used again and again and again. So we have the energy savings of it facilitating this quickly, calling the right players, and then being used again or recycled. So we recycle our enzymes over and over and over again. We save a lot of energy in that way. So we don't need a ton of enzyme within our cells. We only need a little bit. Because they get used over and over and over. They just keep facilitating the same reaction with new substrates to make new products, but they get used again. Enzymes save cells so much energy. The evolution of enzymes has saved living things, and whether we're talking about bacteria or fungus or plants or us or other animals, it saves us a lot of cellular energy. All right, so let's talk about factors that affect the speed of enzymes, because they're not always going to work perfectly, because we know sometimes things go wrong. So let's take a look at all of these ideas here. Okay, so one, this totally freaks me out and blows my mind, that enzyme reactions can be in a cell 600,000 times in a second. A second. Can you do something 600,000 times in a minute? Our cells can do it in a second. So these enzymes are like calling everybody over. I mean, this is crazy, right? I always think, like, if this is happening for one enzymatic reaction, and we have thousands of them happening in a cell at any given time, and we have trillions of cells in our body, why are we just not, like, vibrating with all of this movement? I don't know. It's wild. Totally wild. So they will work at their maximum speed if they have the right amount of substrate available. So as long as there's 600,000 substrates available every second, they will work at 600,000 times per second. But what if they only have 10 available every second? Can they work to that maximum speed if they only have 10? No, right? They do their 10 and then they're like, okay, I need 600,000 to work the fastest. So not having enough substrate will slow them down. If you increase the substrate, let's say that, so here they have 600,000, they can process 600,000 substrates in a second. Let's say they have 1 million available every second. They cannot work faster than this, even though they have way more substrate available. So having too much substrate does not speed them up. They only work as fast as they can go. Just like you. If I was like, okay, your grade is going to depend on if you could go out and run a 4-minute mile. Anybody 4-minute milers? Yeah, I don't think anybody in here. You'd be Olympic bound, right? So it's ridiculous to ask something to work faster than it can work. So as long as they have enough substrate, they work to their maximum. If they have more substrate, it doesn't make them go faster. You gotta have enough enzyme. So let's say that you just had the flu. This was one of the worst flu seasons we've had in a long time. When you get the flu, let's say that you got the flu really, really bad, and you had a high-grade fever, you had 104 fever. What happens in your body is that as your body temperature goes up, that if you kind of remember a little mention of this when we talked about the different biomolecules, was that proteins in our body can get denatured, meaning that they can get broken down when other factors are released on them. So temperature, our body temperature is dependent on a very small range, because when we start getting into 102 plus, that heightened temperature, which is supposed to do two things. It's supposed to give a little boost to our immune system and then also slow down whatever the infectious agent is that's why we get fevers, is to make our white blood cells work faster and to limit the infectious agent. But when you start getting a really high fever, what that means is this infectious agent has been too much for your immune system. And your brain is like, let's kick up the temperature more and more, because you start to kind of like lose it. And what can happen is that you can start to denature proteins in your body. Now, proteins, they account for a lot of the most important things that happen in our body, including catalyzing reactions. And so when you get a high-grade fever, you do denature or you destroy enzymes in your body. You might not have enough enzyme to make reactions go when you get a high-grade fever or the weeks after. So you know when you're really, really sick, it takes weeks, you kind of feel weak and you feel exhausted. That's because your body is going to have to replace all of the things that got broken down and denatured when you were sick. So that's what recovery is about. Recovery is a real thing all the way down to the cellular level. So when you get sick, you do have to slow down. You do have to eat healthier. You do have to drink more water. All of those things are important to getting us back up to speed. So you might get sick. You might not have enough enzyme to make your reactions go as well as they can. So, oh my gosh. So do you think temperature affects enzyme speed? Yeah, for sure, right? Too high grade fevers are you fall into freezing cold water. My son yesterday, he was like, I'm gonna go bike riding. And I was like, okay, make sure you wear mittens or gloves. Do you think he did? No. He came home crying because he could not feel his hands. He got like a mile away, and then he realized he couldn't feel his hands anymore. And they, you know when they just burn, like your hands are so cold you can't feel anything and they burn? Yeah, so by the time he got home, he was like shaking and he was crying, and so I put a heating pad around his hands. And I was like, you know when mom says things like, wear gloves? He's like, I know. Okay, so enzymes are proteins. Enzymes can be denatured. Denatured, think destroyed. Denatured, destroyed. It means that it changes its shape and it changes its function. So when we're talking about any of our proteins, especially like enzymes, who the substrates are reliant on them having a specific shape to bind to, that if our enzyme, which was, you know, in my example here, it's capturing a substrate that's round, well, what happens if it's like this now? It's denatured, it's changed its shape. Can it grab onto that round substrate anymore? No, right? It can't grab onto something like this big when it's like this. So when enzymes or proteins in general get denatured, they get cooked and they lose their natural shape. And then thus, if you don't have the right shape, you can't do your right function. When you get sick, cellular respiration is affected. Your means in which, if you get a high-grade fever and you start to denature enzymes in the metabolic pathway of cellular respiration, of which there are many, many steps within your cell, is you can destroy and cripple the very thing that gives you the little ATP energy to run all of your biochemical reactions in your cells. So getting a high-grade fever, you don't get to break down your food efficiently. And when you don't have enough energy to supply out to your cells, you can't support your immune system well enough. That's just one of many examples, is that you are exhausted, right? You're like trying to feed your immune system army in a way that you can't properly feed it. And so your body's working really hard to do that with the limited amount of metabolic pathways that aren't destroyed. And so you're just tired, you're really, really tired. It takes weeks and weeks to recover to replace all of the denatured proteins in this metabolic pathway. All right, temperature. So one of the things about temperature is that we know, just in general, laws of physics, is that when you increase temperature, it makes molecules move faster. If you go home tonight and you want to make mac and cheese, and you put water on the stove and you release heat energy, that water, you can start to see it move, right? You see the bubbles. That you add heat, it moves faster. So a little bit of heat is good. Low-grade fevers are great. The whole thing about a low-grade fever is that it makes your white blood cells, your army of your immune system, work faster. And most of our infectious agents, they don't like higher fevers. They like our regular body temperature. So they slow down and you speed up. So that's good. But if things get out of control, if they are like, I'm one kind of infectious agent that doesn't care about temperature, and then your white blood cells are like, oh crap, and your brain starts pumping out a higher fever, and then you just like, the whole system goes out of whack, it hurts you. So enzyme activity it's going to decline after enzymes are denatured. They've lost their shape, they've been cooked, they've been frozen. Okay, we're almost done. All right, so your body works best at normal body temperature. If your body gets too cold, your proteins, your enzymes, they don't function as well. If your body gets too hot, function goes down, right? So we want to stay at whatever your optimal body temperature is. They often say 98.6. For me, I'm always at about a 97.1. I operate at a lower body temperature. Everybody has different body temperatures. Too high or too low pH can essentially cook your proteins. Anybody like ceviche? Ceviche fans out there? Oh, I love ceviche. It's one of my favorite things. You use citrus, lemon, lime. You squeeze it on raw fish, shrimp, conch, and the enzymes kill any bacteria and they denature the proteins just like cooking does. And so you can use pH to cook food and it tastes delicious. So at certain pHs, you can denature stuff. Now, most of our pHs, most of our enzymes in our body like about a neutral pH. So like, for example, salivary amylase in our mouth. Salivary amylase begins to break down starches or complex carbohydrates into simple carbohydrates. We keep our mouth at about a pH of 7. If our mouth gets very acidic, what can happen? Like you can get sores in the mouth, right? If you eat a lot of acid. Anybody sensitive to acid? Like me, I love ketchup. I love citrus. My son, kiwi, he ate like 4 fat kiwis last week, and his mouth was like on fire. So too much acid, too much base in our mouth, it can mess things up. It can denature enzymes. It can denature your salivary amylase. But in your stomach is a different story because we have a pH of about 2 because in reaction to food going into our stomach lining releases hydrochloric acid. And so the pepsin enzymes that break down protein in our stomach, they work better at a pH of 2. But most of our enzymes, they like to be about here.
So when you get acid reflux, is that like your stomach acids
Yep.
coming up?
Mm-hmm. Yeah, you have it's called a sphincter, like your butt has a sphincter, your stomach has a sphincter, and you can have issues with that. That makes that come up. Yeah, it's you could have to have your esophagus replaced if you have too much acid reflux. So That is definitely something, if you get acid reflux often, you want to talk to your doctor about it because it can lead to very serious issues. All right. No, let's stop here because this gets into too much and that's going to take another 10 minutes, so let's stop.